Okay, let's take a look at the images that I take under the fluorescent microscope. First one, we started with a violet light, right? And this one is the image from this brain slice. And I'm trying to take photo from corpus callosum. So this is the corpus callosum, and this is under 4x objective lens, and this is DAPI stain, so I use violet light. You see that's violet, and then you see the color coming through, the light coming through, that's violet light. And the emission light is blue, that's why you see the blue here. Now let's go higher magnification. So this is a 40x objective lens. You can see all these cells lined up, which we already see them before. And then this is under a cyan excitation light. So I turn the light into cyan and see the light coming through from this way. That is cyan. And the emission is green. So that's why you see this green here. And this is the GFAP stain, which means all these stained region, they belong to astrocyte. So this image is exactly the same area as the DAPI stain. So this one and this one, they are the exactly same area. So how do I get this image? I just turn the light into cyan, and in the lab manual I said you can adjust focus and camera, but you cannot move the stage horizontally. If you move the stage horizontally, which means you are going to shift your view, your area is going to change. So that's why you cannot. So that will make sure this image will match exactly the same as your DAPI stain. So let's take a look if they match. So you see there is a bubble look-like structure. That's actually the soma of an astrocyte. And you see this line here, the spine line here, that is process. So all these lines are the process, the legs and arms of the astrocyte. And then inside the soma, you're supposed to find a nuclei. Let's see if you can find a nuclei here. Notice where my cursor is. It's right in the center of this little guy. I'm supposed to find a nuclei here, right? Let's see if you can find a nuclei in the DAPI stain, right? So this is the nuclei for this astrocyte, right? So the GFAP majorly stays inside the process of the astrocyte, and something around the nuclei in the soma. Now let's take a look at the DAPI. There is another nuclei here. Once we switch to astrocyte staining, the GFP staining, we don't see anything here, right? And that is because this nuclei probably belong to oligodendrocyte or microglia cell, right? Okay, so this one is still corpus callosum. Under 40 objective lens, excitation light is green, which is like this, and that's green. And the emission light is orangish. However, I don't have anything staining here other than these off-target stains here. Is there something wrong? No. Why is that? Because we are staining the corpus callosum, is that right? You remember what is in the corpus callosum? That's majorly the axons, the fibers, right? There is no soma, neuron soma. So the neuron is staining a neuron protein in the neuron, and particularly this protein stays in the soma of the neuron. So you don't have much soma, you don't have any soma in corpus callosum, right? So that's why you don't have any staining here. Okay, next one is caudal putamen. So if you remember, caudal putamen is right underneath the corpus callosum and laterally to the lateral ventricle. So this is where the caudal putamen is. I use this slice, so caudal putamen is somewhere in here. And first one is a violet light under 40x objective lens. You cannot tell much detailed structure under 4x objective lens, so we have to move up to 40x. Well, what you can do tell is there's a hole here, and that's actually not a hole, that's a physical structure, that's a blood vessel lumen. Why can I say that? Because you see all these dots running along that is blood vessel wall. Let me zoom that in. See, there's dots running around that hole, so that's a blood vessel. Okay, let's take a look at their 40x objective lens. So these are the nuclei we can see under 40 objective lens in caudal putamen, right? Okay, this is the astrocyte stain for caudal putamen. You see there's a more astrocyte staining in particularly in this darker region, and in this dark region. In this region, there's a little bit, and it's hard to find something in here, right? And why is that? Let's take a look at the Nguyen stain. Okay, so in the Nguyen stain, you have this darker region here, there's darker region here, right? Which means in these two regions, we don't have any neuron soma here. And then the major neuron soma stays in this area. Okay, let's figure out what is this black region here. So because our microscope has a white light, which means the light, excitation light can be white. So I use the white light as our excitation light so we can see all the three colors that we stained. Okay, now it's kind of clear. So this is the region that doesn't have any nucleosoma. And you see there's a line of nuclei here, right? And they belong to oligodendrocyte because they're trying to myelinate one axon, right? And then around these oligodendrocyte, you can have some astrocytes. See all these astrocytes? Okay, there's this one here, and its nuclei didn't get stained much because the nuclei probably in another focal plane. In this region, you have another line, 3 lines of nuclei here, and these are the lines for oligodendrocyte. And then in this region, that's majorly where the neuron soma stays. And same thing for this region. Okay, next one. This is the 4x objective lens for cortex. And you see the meninge layer, the bright layer, and then the first layer of cortex. This one I use the violet light. See the violet color? And it is still from this slice. Okay, closer look. This time under 20x objective lens. So this is the meninge layer. You can see even under higher magnification, we cannot differentiate the 3 layers of meninges, right? So that's basically mixed up together. And then the first layer of cortex from here to here. So it's relatively darker. And there is a lot of nuclei in this region. Let's go to astrocyte. So this is the astrocyte stain, the GFAP stain. You can find some GFAP stain in the first layer, which means there is some astrocyte in the first layer. But if you go deeper, the deeper you go, the less astrocyte you can find there, right? And then the meninge layer, you can see a very bright staining. Are they astrocyte? The answer is no. But why do they light up? Because there are some other cells in the meninge that also contain GFAP. Okay, so that is the Nguyen stain. This is excitation light is green, the green light, and the emission light is orange-ish. So you see the orange-ish, that's the soma. Okay, you see in the first layer, there is like a few soma of the neuron, but majorly there's not much soma, right? Okay, next one. This is the hippocampus and the 4x objective lens. So you remember we have this thing tiered apart, but it's okay because we can still take photo of that, right? And then the whole hippocampus stays in this part. So I can take a whole photo of hippocampus in here. And next one, I need the dentate gyrus, particularly the granule layer of the dentate gyrus. So remember, this is the dentate gyrus and this granule layer. So I switched to 40x, and that is the granule layer. And you see there are so many nuclei here, right? And then I switched to cyan light, and this is still the region is the granule layer, and you see there are a lot of astrocyte processes, right? And then this is a Nguyen stain, so there are a lot of soma in this dentate gyrus granule layer. Okay, next one is the CA1 pyramidal layer. So you remember this is the CA1 pyramidal layer. There are a lot of nuclei here, and there are some astrocyte in here, right? And I find this guy here. This is the soma of that astrocyte, and then the other process sticking out. Okay, look, this is the Nguyen stain of the CA1 pyramidal layer. Did you notice other than the soma gets stained, you have a longer axon or dendrite get stained here too? See all these long fibers reaching out? So in these fibers, there's also some Nguyen proteins in there. Okay, so I have some other image that I wanted to show you. So this is the image from previous lab sections. And this one was taken under the white as the excitation light under 4x objective lens. And then I circled here. That is a region that we can see under 20x objective lens, and there's still white light. and then this one is 40x objective. So I want you to take a closer look of this guy here. The DAPI stain was not that strong here. So this bluish region, that's the DAPI. And then in this image, you can see the soma of the astrocyte very clear. See this soma here? And inside this soma, you have some bluish staining, that's the nuclei. And then this is the neuron soma, that's fairly big compared with the soma of a astrocyte. Is that right? Okay, so these slides are already pre-made. You just need to take a look at them under the microscope. First thing, I need to put that slide under the microscope. By holding this clip, I can slide my slide in and hold that in there. And then I need to make sure that my bright field light is on. In this case, it's already on. The button is turned into 1, that's on. And then I can change my light intensity by turning this knob. And if you see, I turn that off. There's not going to be light coming through. So I have to make sure that my bright field light is turned on and my fluorescent light is off, because I don't need my fluorescent light for this, right? And then next thing, we have to make sure this lid is not covering our bright field. If you put your lid here, there's no light coming through. So you have to make sure that this lid is not blocking our way. And we start with the highest position of the stage. We start with 4x objective length. Now you're ready to search for your bisecting neuron under the eyepieces. And once you find it, you can take photo on your camera. Okay, so this is the image of this bisecting filled up pyramidal cell. So this is the whole brain slice. And then your target is very small. It is this little guy here. I'm going to blow it up. See, this is the one. That's somebody use a very fine needle to inject bisecting in this neuron. So that's a really fine work to do. And did you see how small it is? So when you're looking for this cell, you got to be use 4x objective length. Otherwise, you are going to search a lot of miles to find this little guy here. Okay, so this is the target that we need to take a closer look. And if we blow up to 40x objective length, it looks like this. Early on, I told you the axon of this pyramidal cell have a terminal, right? So this is the axon terminal. And then this is the soma, and then the opposite side of this axon, this is the apical dendrite. So you see the apical dendrite will go all the way to the surface of the brain. And then all the dendrite, the same side of the axon, they're basal dendrite. Okay, I have more image for different cells. So this is the one that we already talked. And this guy here, you see this is soma, this is axon, this is terminal, and all the apical dendrite, they're here, and the basal dendrite here. And same for this one. So he has a clear axon terminal, and this is the apical dendrite. So it's going to go to the surface like this. I mean, some of them probably will not reach the surface, but it's reaching the surface direction. Okay, and this guy here, the soma is in the another focal point. That's why you didn't see that clear. But the axon, the terminal, looks pretty clear. And the apical dendrites are these dendrites and this big dendrite here. Like other, pretty much they all have this big apical dendrite. And then the basal dendrite at here. Okay, so that is all about the microscopic brain examination. I know it's kind of hard for most of you guys because you probably never seen this before, and that require a little bit spatial thinking. So if your grade is not ideal, don't feel frustrated, because that's just the way it is. I know it's hard, and if you keep your hard work, you are going to get a good score in the end.